Hello, my name is Autumn Berlied, and today I'm discussing my master's capstone, a bioinformatic approach to biomarker and target discovery in uveal melanoma. Current research suggests that within populations of Europe and the United States, there are approximately 6 cases of uveal melanoma per million individuals. And of these impacted individuals, nearly half will develop high morbidity metastasis. Despite rare occurrence, uveal melanoma is too frequently a precursor to metastasis and death, prompting the necessity for advanced tools that reach beyond the scope of standard diagnostics. In addition to rare occurrence rates, such malignancies can be clinically misdiagnosed as benign conditions of the eye or adnexa, delaying crucial treatment time. The site is exceptionally sensitive, requiring the use of specialized technologies and forcing medical providers to manage the priorities of minimizing vision loss, disfigurement, or even death from the malignancy. For these reasons, standard clinical procedures often fall short. However, this gap is being filled by new and emerging technologies which elucidate on the unknowns of this rare and deadly cancer. When evaluating the TCGA UVM dataset, I had a few questions in mind. The top 5 were this: what are the top mutated genes associated with uveal melanoma? Is the TCGA UVM dataset representative of populations with this aggressive malignancy? Which associated genes are the most poorly misunderstood? How do gene expression levels associate with survival? And which of these genes are potentially druggable? GNA11 encodes a signal transducing G protein subunit that regulates melanoblast migration within the dermis. Recurrent mutations in this gene have been widely documented as drivers behind uveal melanoma. Next, SF3B1 encodes for a crucial RNA splicing factor, and mutations allow for its behavior as an oncogenic driver. In contrast, BAP1 acts as an essential tumor suppressor gene, regulating chromatin remodeling, cell cycle progression, and apoptosis. COL14A1 is not well studied in the context of cancer, but it is documented to regulate fibroblast apoptosis. Consequently, these 4 genes will be of primary interest for the purposes of this study, with COL14A1 in particular because of the noticeable lack of research concerning its role in uveal melanoma. Throughout the process of this study, I started with a literature review. I wanted to see what genes, if any, were known to be associated with uveal melanoma. Next, I wanted to do an R analysis of the TCGA UVM dataset that I queried from within RStudio. From here, I performed mutation and SNV analysis, DNA methylation analysis, survival and genetic alterations analysis, and I even was able to make a druggable categories figure and query a database. Finally, after all of that, and I got the genes that were mutated in my dataset, I was able to conduct another post-analysis literature review and determine which genes might be best for further interrogation by other scientists down the road. So like I mentioned, I used RStudio for my analysis, and I used a lot of different packages. I used Dippler, Survival, SurvMiner, Complex Heat Map, TCGA BioLink, Summarized Experiments, ggplot2, Tidyverse, MathTools, Sesame Data, Sesame, Genomic Data Commons, pHeatMap, and DESeq2. I also wanted to note that only open access cases were utilized for the purposes of the study. Further interrogation of restricted cases would need special permissions, with which I, the author, was not granted at the time of writing this article. Using GDC query and MathTools, I was able to create 2 mutation and SNV, or simple nucleotide variation figures, and this is the first one, which is a multi-panel figure. You can see A is variant classification, B is variant type, C is SNV class, D is variants per sample, E is variant classification summary, and F is top 10 mutated genes. Just looking at this more broadly, we can see that the most common variant classification is the missense mutation. The most common variant type is the SNP. The most common SNV class is a switch from a C to a T. And then we can see that variants per sample mean is around, or the median is about 10 variants per sample. Classification summary is basically just another visualization of the types of classifications of variations. So again, we see that there are more in the missense variety. And then finally, I think the most interesting part of this figure is the top 10 mutated genes. And we can see that GNAQ or GNAQ and GNA11, which were found to be the most commonly mutated genes in uveal melanoma cases in my primary literature review. It was interesting to see that my data set, or the TCGA UVM data set, was actually representative of that, and they were the most two common mutated genes in this data set, followed by SF3B1, BAP1, and at the bottom there, my last gene of interest, COL4A1, which was mutated in about 4% of the cases. Now, this is my second mutations in SNV figure. It's basically a more detailed breakdown of figure 1F from the last slide. And we can see that there were mutations or SNVs in 97.5% of the samples of the cases in the data set. So 78 out of the 80 samples in the data set had mutations or SNVs. And we can see that GNAQ or GNAQ was mutated in 49% of the cases. And primarily it was of the missense mutation variety, but there do appear to be some that are multi-hit, and then followed by GNA11 in 44%. Those are all missense mutations. SF3B1, all missense mutations. BAP1 it gets more interesting. There's more frame shift, missense, and we also got some nonsense. And so we have both types of frame shift, deleterious and inserts. And then finally, you can also see that COL14A1 was also mutated in about 4%, and they were all of the missense variety. And on the side, it just shows you like a number of the actual samples instead of just a percentage, which is on the left, which is a great visual breakdown of all the mutation data in this data set. Normal healthy cells have proper regulation of gene expression and gene silencing, whereas DNA methylation, either hypo or hyper, is associated with histone modifications, which ultimately act as regulators of the genome. Methylation beta values range from 0 to 1, and the value represents the average methylation within that sample, where a value closer to 0.0 denotes unmethylated or hypermethylation, and 0.5 denotes partial methylation, and a value closer to 1.0 denotes fully methylated or hypermethylation. Methylation beta values for this set were moderate. The mean was actually 0.518, with a heavy clustering around moderately hypermethylated beta values. To better understand the implications of specific gene changes on patient survival, survival analysis was performed. Cases with higher or lower raw counts than the median were tagged as strata accordingly. And so for GNA11 in A, you can see that individuals with high raw counts of the gene had increased survival over time compared to individuals with the lower raw counts. P-value 0.017. Conversely, for SF3B1, individuals with low raw counts of the gene had increased survival over time compared to individuals with higher raw counts. P-value 0.014. For BAP1, individuals with high raw counts had increased survival rates over time compared to individuals with low raw counts, p-value less than 0.0001. And finally, for COL14A1, individuals with low raw counts had increased survival rates over time compared to individuals with high raw counts, and the p-value for that was 0.022. The survival versus genes of interest analyses revealed only one relationship, which was statistically significant. That's the BAP1. And patients here with higher raw BAP1 counts had increased survival rates over time than patients with lower raw counts. This finding is consistent with the literature, which states that this is a crucial tumor suppressor gene, and increased survival might be expected when the tumor suppressor gene is transcriptionally active and the gene is able to perform its role effectively. Finally, for the druggable categories, I was able to make this figure based on the top implicated genes in the cohort. The primary category results in the druggable genome and the clinically actionable for genes which were previously described in the first set of figures, the mutations and the SNPs. These genes include BAP1, EIF1AX, GNA11, and GNAQ. Some genes are listed in multiple categories as they fit into several different groups. And genes in the druggable genome include BAP1, C3, CA, CNA1D, COL14A1, and CYS LTR2. Looking at the druggable categories and querying the DGI database, which is the drug gene interaction database that I queried from within RStudio, I was able to determine drugs that are currently approved for specific gene changes, and it was revealed that SF3B1 and BAP1 and GNA11 had approved drugs. Additionally, the querying showed that COL14A1 did not have any currently approved drugs, whereas GNA11 had a dozen targeted therapies. Although COL14A1 had no targeted therapies at the time of writing this, it was depicted as a druggable gene. Unfortunately, the rare occurrence rate of uveal melanoma in combination with the mutation rate of this gene for this dataset, it may dissuade pharmaceutical companies from developing such a drug for COL14A1. Gene mutations are a primary focus of innovative cancer research. Although gene mutations are often associated with cancers, particular genes associated with uveal melanoma, which is a high mortality cancer, are still poorly understood. Using bioinformatic techniques and an open source database, I was able to examine several genes implicated in the rare but deadly uveal melanoma. The findings here suggest that the selected genes are valid therapeutic targets that may be effective if invested in, despite anticipated minimal financial returns on such a product. And finally, for my simple summary, genomics and gene expression are key players in cancer research, yet these avenues remain largely underexplored in uveal melanoma. In this study, 4 genes of interest were selected and more thoroughly explored, confirming previous findings and suggesting new routes for further investigation. Finally, the genes selected were all determined to be druggable, despite rare occurrence rates dissuading pharmaceutical companies from investing in R&D. These are my references. Thanks for watching!